Thank you all for joining me today. I'm going to be talking about doing more with less, maximizing phase-appropriate DOE strategies. And hopefully by the end of the day, you're not thinking that that means do the minimal amount at each phase. So let's go through an overview of what we're going to talk about. First, I'd like to introduce Lundbeck in case you haven't heard about us yet. Then walk through the phases of CMC development and how we might maximize the efficiency at each step using our DOE toolbox, what, when, why, and how, and then understand the analytical variation to set ranges as part of our pre-DOE homework. Then I'm going to take a project and walk it through early development and late stage development and what we would do in DOE at each of those phases, and then talk about a relatively new auto-validation strategy that harnesses the power of machine learning for small data sets to analyze that late stage development definitive screening design that you can then use for de-risking your testing the model and going and advancing your process development. So who are Lundbeck? We are a global pharmaceutical company specialized in discovering and developing innovative treatments for brain diseases. Our goal is to be #1 in brain health. And if you are interested in learning more about us or applying for any of our many open positions, please read more about us at www.lundbeck.com. We have over 5,000 employees in over 50 countries. Our largest stakeholder is the Lundbeck Foundation, which allows us to focus on the brain diseases that we want to help cure. Our 2020 revenue was over 17 billion Danish kroner. We treat more than 7 million people per day with our products. Lundbeck was founded in 1915 by Hans Lundbeck. And in 2009, we established a U.S. presence. In 2019, Lundbeck acquired Abide Pharmaceuticals, a small molecule research company in La Jolla, and Alder Biopharmaceuticals in the Seattle area that was working on antibodies. And Alder was where I was working. Our focus is transformative medicines for niche and rare diseases, and we run the gamut from early research for drug discovery through drug development, product development and supply. We have corporate support, and that's supporting our global presence. Having all of the functions for the drug development process means that we have an opportunity to interact with stakeholders every step along the way, which I think is important as we strive to meet patients' needs. Let's now talk about the phases of CMC development. So after your research and development group have defined a product that might meet a patient's need, then it gets transferred to the chemistry, manufacturing, and control section. And as we start along this pathway toward launch to get the drugs that they need, it's going to increase your resources, your personnel, and your cost at each step of the phase. So treating the patient is the goal. How can we balance the effort and resources to maximize the speed and efficiency of drug development as we walk along that development timeline? When we're in the first step with our first little diamond here, we're working on developing the first-in-human process. This does not need to be the final process at this stage, but we want to still have a process that is safe and for the patient, and we can look at how efficacious it is. In this case, for the upstream development group, we're going to focus on choosing a robust cell line so that we can maximize the efficiency of that timeline to get that product in the clinic if we can. If we don't have to do a cell line change, that makes life easier for our quality folk, our regulatory folk later on in the process, as the cell line change is a major change, and we would want to do extra work to make sure that that change is still safe for the patient. When we have that first product going through phase 1 clinical trials, that's when we want to initiate our commercial process development. Here we want to optimize for yield and product quality because we want to develop the process that's going to be used for phase 3 clinical studies. The process that's used for phase 3 clinical studies should be the same process used for commercial launch. Typically, after we've developed that process is when many companies start the process characterization in order to have greater understanding of the operating space, greater understanding of your process. That's when a lot of time and effort is spent on running experiments to establish that understanding for safety, again, for the patient, with the goal to meet a validated commercial process that can then be launched. I'm hoping to show you today there are steps that we can do in step 2 that may make the step 3 more efficient and that you could leverage for a quicker time frame. And one of the ways that we're going to do that is use our DOE toolbox. So what is design of experiment, and why would we want to do it instead of using one factor at a time or OFAT studies? Design experiments are experimental strategies that are mathematically designed to extract the maximum information from the least number of runs. When do we use them? There are different types of DOE strategies. So I truly think of DOE as a toolbox. When we're early in development, I'm going to use screening designs. In this case, I'm focusing on the factor that I'm changing, or think of it as a knob that you're turning. Which of those knobs are the most important for your process? And so that's, in our case, we're looking at titer or yield and product quality. When we get to the later stage, when we have developed our commercial process, that's when we really want to focus on characterization. And we want to predict an outcome depending on what that factor or parameter setting is within that design space. And process understanding for late stage development and even post-launch. Why would we want to do this? We want to gain understanding of parameter interactions. For instance, if you're baking cookies, you could probably understand that there is an interaction between the time that you place the cookies in the oven and the temperature. If you have the temperature too high, you might be too crispy on the outside and gooey on the inside. And likewise, if it's too low, you might have to increase your time. So having an understanding of those parameter interactions, how would that affect us? We might, if you try to maximize or optimize a strategy one factor at a time, you would be optimizing, say, for pH. You would find that perfect pH while keeping all other factors constant. But then when you try to optimize for temperature, what if you were at the wrong pH for that temperature to meet your highest yield? If we could do that at the same time, you'll maximize your speed of efficiency to develop that process. So we want to gain that understanding and also do it in a time-efficient process. Our little schematic in the bottom right just shows, each dot shows how many runs are going to be needed. And in this case, this is just three-dimensional space or two or three factors, but can you imagine how many factors that are affecting your process can be much higher? Now let's talk about how you would do a design of experiment. First, collaborate with your stakeholders. Remember to keep the patient in mind. If the research folk can talk to the CMC manufacturing and find out what is needed to speed process through development. In one case, we had an antibody coming out of research that was sticking to all of the tubing that was used for the product quality for analysis. We quickly determined that that was not a very manufacturable antibody and switched to the number 2 in line, and that has gone forward and been launched into a product. Let's determine the timeline and resources available. That's going to help you determine what type of DOE strategy you're going to use. Identify your parameters to investigate. So have an understanding of the reproducibility and the standard deviation for your process. It can be for a prior project, but you need to have that understanding before you set those ranges. When you do set those ranges, be brave, go wide to ensure higher signal to noise ratio. You don't have to try to hit your best conditions in the experimental design. You're hoping that the mathematical models will be able to predict what is your best condition at the end of the study. So pick the model that fits your goal. Are you going to do a screening design, or are you going to do a characterization design? Characterization designs are usually done with fewer parameters, but there's more runs so that you have really understanding of that operating space. Many software are available now that can harness the computing power for modern designs. I use JUMP. Full disclosure, I am on the JUMP steering committee. That is the program that has worked well for me, and I like many of the designs that are coming out from the developers that work there. Now we're ready to execute your experiment. Complete your experiment in random order. That is very important. If you need to talk to the operators on the floor, if there is a particular parameter that is difficult to change, you want to include that in your design so that you can block by that parameter. Now we're ready to do the fun part, the analysis. For definitive screening design, Brad Jones has created the FIT definitive screening platform, and that is available in regular JUMP. What I'm going to talk to you today about as well is a new strategy developed by Chris Gottwalt and Phil Ramsey, which is an auto-validation strategy that's using machine learning for small data sets. This particular strategy will require JUMP Pro. The benefit of using the auto-validation strategy is it de-risks that final step of DOE, which is to test your model. Remember George Box. All models are wrong, but some are useful. In our industry, every run is expensive, can be expensive, and so you want to speed up and make sure that the time that you're taking to test your model is the most practical and has the highest chance of success. And using the auto-validation strategy has worked well in my case. Now we're ready for our pre-DOE homework. So this is one project that we finished, and I've used this project to have some understanding of the equipment that I'm using. The graph on the far left, the titer graph, is showing a titer profile over time. The x-axis is showing the culture time in days, and the y-axis is showing the BioHT normalized IgG titer. And again, thank you to Roche Custom Biotech for the invitation to speak today. The BioHT is one of my favorite pieces of equipment, and we are very happy with its reproducibility and consistency. But in this case, we're also measuring what's the consistency between different bench scales for bioreactors. We have the 250 ml amber, we have a 3-liter and 14-liter single-use vessel. And we're seeing nice reproducibility between scales, between users, and between run sets. On the right side, I have a product quality from a analytical reference standard that's been in the middle graph is overlays of a capillary isoelectric focusing graph. The peaks that are to the far right and far left peak are markers. The peaks that are to the left of the tallest peak are considered acidic species, and this is a product quality parameter that we investigate, especially early in development. It's a measurement of how your protein can change over time, and it's especially impacted by pH, time, and temperature. So for this reference standard, I want to know before I get into a designed experiment, what is the variation in the analytics? What's the if it's run between different instruments, between different reference lots, between different analyzers. On the far right graph, I have the shaded area that is showing the range that we're seeing for acidic species. This has been, the mean is normalized at 1. I need to go back to my analytics group and just double check and say, is there something special about the outliers that are lying to the bottom of that mean? Or is this true deviation that I could be seeing in my experimental design? Then I want to make sure that my ranges are wide enough that my signals to noise is going to be higher, and I can make informed decisions going forward in my modeling. Now let's talk about setting factor ranges. I've been doing designed experiments for a long time, and I have had a lot of requests for doing multiple levels of an experiment. And for this project, I'm showing to the far left the full data set where we tested multiple pH conditions. So in this case, it's just one factor, multiple levels. In the end, we found the optimized setting, which is in blue. But let's take this as an example for how we should set ranges during a DOE. The next graph over to the right is using a conservative range setting. In this case, the middle points that were tested were the platform process conditions. And it looks, if we were using that conservative range setting, that the platform conditions were the best conditions to go forward with. And in fact, if we went up in pH, we might be getting into a dangerous spot, but it looks pretty safe for where we're at. But you'll notice that we didn't quite hit the optimized setting. If we had used a wider range, then we are able to predict mathematically the optimized condition that turned out to be the optimized condition. And we also note that if we drop in pH, we're starting to see a significant drop-off in our titer yield. We get that same information with a bold range setting. And and again, this is just 3 levels. We're still able to see and determine mathematically what the optimized pH setting should be for this process. So I encourage you, do not need to test every level in between for a parameter that you're setting. And this is one of the powers of DOE. Now let's get into our case study. We know that our equipment's working. We know what the settings that we need to do, and we're going to be bold in our range setting. For this project, we start with early stage development. So we're taking from our cell line development group, they're screening the top 12 to 48 cell lines in shake flasks, and they identify the top 4 to 8 cell lines to go forward for bioreactor screening. In this case, our filters to decide if a cell line is going to go forward for processing are yield, product quality, and manufacturability. And then another key point to look at is stability. If this cell line is passaged for multiple generations, does it still maintain its genetic stability? Is the product, once it's put into gone through the purification and it's in its final formulation buffer, is it stable over time at the temperature that you want it to be? The stability aspect of the filter is going to, by necessity, take time. But for manufacturability, we can be poking at that a little bit earlier. Again, if we don't have to do a cell line change after phase 1, we can save time and resources in the company for not doing a major change. So we're going to talk more about manufacturability. So let's explore manufacturability with the top, in this case, 3 cell lines. I usually take the top 2 or 3 cell lines. We want to balance the desirability of the response with the robustness across a moderate process range. So in this case, I'm not being very bold with the pH range settings that we're doing. If you look at cell line A, it had the highest titer from the previous slide when it was run under the platform process. But it has the lowest manufacturability. What do I mean by that? When you do minor changes in pH, we see a significant drop-off in titer. Cell line B had the lowest titer, but it was fairly robust across the same moderate pH range. Cell line C had a little bit of movement with our pH range that we were testing and high titer. So that's the cell line that we're going to proceed forward with for development. And again, this is early stage development. We don't need to have the final process here. We just need to know which cell line is the best one that we want to continue forward while the product is going through the phase 1 clinical trials. So while that's in phase 1 clinical trials, we're taking this project through late stage development. So let's talk about case study number 2. So let's perform a risk assessment before we start doing the upstream late stage development. And we want to avoid prior assumptions. This is a new cell line, it's a new antibody. Let's bring all of the parameters to play that could be affecting the responses that we care about. In this case, the definitive screening design is chosen for optimization because there's many parameters or factors that could be affecting the responses that we care about. The definitive screening design is chosen because it's very efficient use of resources for investigating 4 or more parameters. Not only that, it investigates the low, middle, and high setting for continuous factors. This allows fitting of a curve or having your quadratic model terms. You can include 2-level categorical factors. For instance, if we had 2 different media types that we wanted to include, it's also, you can do blocking effects. For instance, if you don't have enough reactors to run all in one set, you can block by run sets. If the resources are available and you have the time, you can add runs by a factor of 4 to increase your power. These are the definitive screening design was discovered by Brad Jones and Chris Noxheim. And so I have some references here. I also have reference to the JUMP user community with a blog from Brad Jones on the proper and improper use of definitive screening designs. This blog I go to regularly for help in describing to other people the power of definitive screening designs. So we met with all of our teams. We decided what the top 10 factors are that we're going to use. In this case, we needed blocking because we needed to run it over 4 run sets. So we checked the N-1 seed density, the production tank seed density, starting temperature, the pH setting, the temperature shift, the day of the temperature shift, the glucose target level, the feed A and the feed B concentration, and the timing of the feed start. With all of these 10 factors, we can investigate this in a mere 24 runs. And each of the factors that we are testing, the results are not biased or confounded by each other two-factor interaction. What does that mean? The seed density is not, we're going to be able to understand the importance of the seed density on this process by itself, and it's not going to be influenced if there happens to be an interaction between pH and temperature. So this is a very powerful aspect of this design, and that is shown by the color correlation map that you see in the middle there. So here are our results. In blue, we see the definitive screening design results of those 24 runs that we just described. In red is the platform process. In the top left graph, we're looking again at the bioHT IgG titer over time. And you can see that we're seeing from, this is the normalized titer. So we're seeing very low titers all the way to the up to 100%. We didn't find a condition in the DOE that is much higher than the platform process conditions. Again, that's okay. That's not the purpose of the DSD. We're hoping that we'll be able to model conditions that we haven't tested yet that are going to increase our titer. For growth, we're also seeing some conditions that severely limit growth, some conditions that we do have higher growth. That's not necessarily a good thing, but we now know from this range of responses that I'm definitely going to be able to find factors that are very important to yield and important to growth. The right two graphs on this slide are product quality. So on day 14, we take a sample, we purify it through the first purification step, protein A capture, and then we sent it off to analytics for analysis. The top right graph is the results from size exclusion chromatography, and we're looking at aggregates. If there's a lot of aggregates in your process, that could potentially be a product quality risk for your patients. In this case, I'm plotting against the platform process results, and you'll notice that the range that we're seeing for the platform process is very similar to the range that we're observing with the DOE. Now, that doesn't mean that the DOE can't predict, because we did have great prediction for size exclusion aggregate values, but what it does tell us is that for size exclusion aggregates, this process and cell line are fairly robust. We're not seeing a lot of wiggle. For the charged species from the CIEF acidic assay that we're looking at, the red platform process results range from about 0.5 to 0.6 of the normalized acidic peak. But for the DOE, we're seeing from 0.2 to 0.9. We're seeing a very wide range, so I definitely have some factors that we tested that are impacting that acidic peak. And we're definitely outside the variation range that we saw with acidic peak. So I can be sure that my signal to noise is quite high. So now we have our results. Let's go and analyze them. In this case, I'm using JUMP. Everyone has this available in regular JUMP, and using the FIT definitive screen platform that Brad Jones created. And how does this analysis work? It's a three-stage process. In the first stage, it looks at the main effects, or the top 10 parameters, and it says for all of those top 10 parameters, how much do each of those parameters changing affect the response of titer? In the second stage, we take the residuals, or the remaining information that is not explained by those top 10 factors changing, and it says, okay, how are the interactions working, or how are the quadratic terms? Is there curvature within any of these parameters that we're seeing? And then the final stage is it glues those stage 1 and stage 2 together to come up with your mathematical model to describe this response. I'm showing in this option 1, you'll notice that the quadratic terms obey strong heredity and the interactions obey strong heredity. Thus, those boxes are both checked. What does that mean? That means if the main effect is not chosen, or the main parameter by itself did not have a strong response on the titer, then it would not be included as in a curvature term or in an interaction with any other factor. And so this is a very simple model, and you have less chance of overfitting your model. The bottom right graph is a Pareto plot, and that shows us the importance. It's a way to show the importance of each model term, which is, think back to your algebra, it's just one part of that equation. So in this case, starting pH is highly important. The next term, model term that it found most important, was a curvature between in the temperature shift. And then it found curvature in pH being similarly important to the temperature shift. Then temperature shift as a main effect. And then it found a very slight interaction between the starting pH and the temperature shift. And in this case, the block was important. So that means there's something that I am not aware of that was changing between block and block. Maybe block run set to run set. Maybe the media lots, there were slight variations. So there's something that I could do to tighten up my reproducibility between blocks. Let's go to option 2. Again, I take George Box to heart. All models are wrong, some are useful. So I like to look at multiple models and get a feel for my data. In this case, we're going to say the quadratic terms don't have to obey strong heredity. So in other words, there might be a case where there's curvature for a parameter that is more important than that parameter by itself. So if we look at the results here, starting pH is still the top factor, and it's in the Pareto plot, followed by temperature shift. So this model is still picking out the same top 2 factors. But now we're starting to see that the production seed density has some curvature to it, as we might expect. Also, there's some curvature that was been found with the feed B percentage. So in this case, we have a few more model terms that have been included. Let's continue on to option 3. In this case, we're going to go back and say that the quadratic terms obey strong heredity, but the interactions do not. So in this case, there might be an interaction, or there actually is being pulled out, an interaction between the starting pH and the feed B percentage. Also, we're bringing in the nutrient feed start time in hours and feed A with some interactions there. Feed A appears to perhaps have an interaction with the temperature shift, etc. Our top factor, again, is the curvature term for pH, and then pH is the second term, most important term for this model. So each model is still finding pH as being the most important model parameter that we tested that is affecting titer. Option 4, this is my favorite. You're not supposed to have a favorite. This is my favorite. I like to have all possible combinations. So I don't have any restrictions on the quadratic terms or the interaction terms. In this case, we have feed A, we have the N-1 seed density, and there's some curvature there. We have some interactions that are coming on, but the starting pH is still the most important parameter. So is there another model we can use? Yes, in regular JUMP, there is also a method where we can use the stepwise forward method using the minimum AIC corrected. I also like to look at this model, and I'm showing what I'm depicting here in these graphs are how you would do that. So if you take your 10 factors and you add them to your model terms as a response service, so that means that you take all your 10 factors, every possible interaction between those 10 factors, and all of the quadratic terms for each of those 10 factors. That's what the response surface model is. And then I add to the Y the response that I'm looking for. In this case, it's the normalized IgG titer. And then you start going forward with the minimum AICC. I am not a statistician, so if you need more information on that, I would point you to the JUMP user community to have an understanding of how this model works. In this case, we are now finding that the glucose target has some curvature that may be affecting our titer. But again, the starting pH setting and the temperature shift are the two model terms that are at the top of our list. So another way to look at this is in a color map. So all of the 5 models that we have looked at chose pH and temperature shift as being the most important factors out of the 10 tested. The simplest model, the option 1 with heredity enforced for interactions in quadratic terms, only found pH and temperature shift. The other models found varying combinations. What is the best model to use? And again, you want to make sure what you're testing to go forward with, or your decisions to go forward with, that your model is going to predict that. So how can we de-risk that next step or that decision to go forward? One way is to use a new method developed by Chris Gottwalt and Phil Ramsey, using auto-validation for small data sets. So using auto-validation method for small data sets, machine learning methods have revolutionized the analysis of big data sets. You can use a fraction of the data for training or building the model. Let's say we use 70% of that data for training. Then you use 20% of that data for validating that model, and then 10% for testing the model. That can all be done in silico. That method is not easily applied to designed experiments. Each run in the experiment is carefully configured mathematically so that you get the maximum amount of information. I'm going to refer you to Chris Gottwalt, who is a JUMP developer, and Phil Ramsey. They gave a talk in 2018. You can follow the link here to see the full talk. I'm going to walk you very briefly through what they came up with and what I have used successfully, and I'm going to show you here for this project. So the setup for the analysis. You're going to create a copy of the original DOE data set and add a validation column. So the original data set is going to be your training set. The copy is going to be used for your validation. You're going to create a new formula column for the fractionally weighted bootstrap method, and that is the formula that I'm showing to the right. Then you're going to create a new column for the null factor. In this case, the null factor is just a randomly distributed result that's independent of the monitored response, such as titer. There is a wonderful add-in, if you have JUMP Pro, that I will refer you to that does all of this automatically. And you can find that in the link shown. What does this look like? The original data set is shown in orange as the training set. We just make a copy of that. We have that for the validation. When you're using JUMP Pro, you would put your Y response, in this case titer is what we're looking for in the Y. You would put the paired fractionally weighted bootstrap in the frequency, the validation column in the validation, and then you would do a full response surface as we did for the stepwise. In this case, you're going to add that null factor that's just a random generated column, random number. That random number is going to change for each time you run a simulation, because now we're going to use the machine learning. In this case, I like to use lasso or double lasso for small data sets, and I'm going to perform 2,500 simulations. In each simulation, the null factor is set to a new set of random numbers. And in order to do that, you have that. I'm showing this how you would set up that simulation in JMP Pro. After it completes that 2,500 simulations, which takes a little bit of time, then you can identify the percentage of time that the null strain was randomly included to be important as a factor. We know that the null factor is not important. It's just random noise because we generated it to be that way. So in this case, every model term that is included less times than the null factor, which is random noise, we know that that is not important. And those are shown in gray. The null factor response is shown in red. To the far left, we see that pH setting and temperature shift have been included in more than double the amount of models of those 2,500 simulations than the null factor. That matches what we knew already, that pH and temp shift are highly important. But now we have some blue factors that they've been included slightly more times than the random noise of the null factor. So if we add that to our model and we look and see what the results are, the starting temperature has been found as being blue, feed start time, and N-1 seed density are three additional factors to pH and temperature shift that might also be important. I will tell you that with follow-up experimentation, starting temperature was found to be important. And only option 2 that we had looked at before found starting temperature as being very important. Still, now let's test the model. Once we've de-risked the final conditions that we need to do, let's perform that test in the wet lab. We used this auto-validation method for each of the factors that we had, each response of interest to us. So growth, viability, yield, all of our product quality parameters. Each response receives its own individual model, and you can save that prediction formula to the data table. To the right, I'm showing the predicted values for the optimized model. The upper green band and the lower red band are showing the upper and lower 95% confidence interval. The blue bar is showing the actual prediction, and the two black dots are the duplicate wet lab results that we used to test the model. So we had a model that worked, and we increased our overall yield by over 20% from the original platform process conditions. In addition, we have a lot more information about the product quality parameters that matter going forward. So in summary, prior to starting DOE, have some understanding of your process variability all along the path. Understand how bench equipment has scaled to manufacturing for past products. Be bold in setting your process ranges. Increase your signal-to-noise ratios to create more useful models. Maximize your efficiency of the CMC development through the phase-appropriate DOE strategies. This requires iterative communication with your cross-functional stakeholders. Balance your resources in a phase-appropriate manner. Thoughtful designed experiments can increase your overall speed of development. Focus on the manufacturability in early stage. Don't try to find the best process. Then you can identify and minimize possible future roadblocks, such as a cell line change. Large DSD DOE studies in late-stage development can be leveraged for increased efficiency of process characterization studies needed to establish a commercial process. Using the auto-validation with fractionally weighted bootstrapping provides useful prediction models from screening designs, such as the definitive screening design. I have lots of acknowledgments for the team that was responsible for creating this work. Our upstream development team that had been led by Mike Salama, our downstream led by Jeff Lee and Ethan Oyla, our analytical team led by Pei Key and Mary Jo Phoenix, and especially our scientific computing with Kira and Pat. Being able to access your data very quickly and look at it across groups, that has been fabulous. Thank you, and do you have any questions?